everyone. Good morning, good afternoon, good evening. Welcome to another edition of Global IMMUNOTALKs. My name is Laura McKay. I'm from the Doherty Institute in Melbourne, Australia. And on behalf of the 2022 Global IMMUNOTALK Organising Committee, it is my absolute honor and pleasure to have with us today Kazuyo Moro from Japan, who'll be our Global Immuno speaker for today. So just before I do an introduction for our Global Immuno speaker, just a little housekeeping, just to remind everybody that questions for the talk will be over Twitter. There will be a Q&A, and this is a pre-recorded talk due to the time zone change with Japan. So those questions will just go on for a little bit of time, not necessarily live directly after the talk, but please jump online and ask your questions afterwards. So next week's speaker will be Ansuman Sapathi from Stanford University. But today, the main event, we're so excited to have you, Kazuyo. Just to give you a little bit of background, Kazuyo is a professor and head of laboratory for innate immune systems at Osaka University, and also Riken in Yokohama, and also holds a position at IFREC. She obtained her PhD in 2007 and absolutely catapulted to superstardom, perhaps best known for her discovery that she made in Riken under the guidance of Shigeo Kasayu on the discovery of group 2 innate lymphoid cells, or ILCs, which she'll be talking on today. Since starting up her laboratory, or I should say multiple laboratories, which is more than most of us cannot cope with once. So I'm blown away by managing double laboratories, I have to say. She has made immense contributions since starting that group in 2015 to our understanding of the development and regulation of ILC2s, with a view to contribute to new therapies against allergic disorders. So thank you so much for joining us and presenting your research freely across the globe. As you know, as part of Global IMMUNOTALKs, we love to get to know you a little bit better and give the audience just the opportunity to hear something personal from you. And so the question we'd love to ask you is, what is the trait in your personality that has most helped your career?
Okay, thank you very much, Lola. And first of all, I would like to thank this opportunity to give my talk in this great
program initiative.
Yeah, private program. Yeah, so I'm very happy to be here and to answer your question. I think I need to talk this funny story about what happened in my childhood. So when I was a kid, my elementary school teacher called my mother and said, "Kazuyo, disrupt class." And my mother was so shocked with this comment, and asked him, how is Kazuyo disrupting the class? And he said, "Kazuyo asked too many questions." So yeah, I have a character that when I have a question, I cannot move on until I got the clear answer. So before I start working on research, this character made many trouble. But now I think this personality is helping me a lot, because I believe that questions are the most important factor to create the originality in research. So maybe that's the answer.
I love that answer, always asking questions. It's just the epitome of being a scientist, isn't it? Thanks so much for sharing that story with us.
Okay.
If you could share your slides with us, that would be great. We're so looking forward to your presentation.
Okay, wait a second. Okay, can you see my slide?
Fantastic.
Okay. So today I would like to talk about the discovery of group 2 innate lymphoid cell, because I have been asked how did you find ILC2 several times in the past 12 years, especially from the young generation, like who are watching this global immune talks? So I'm going to talk how we discovered this cell. So this is my lab member. I have two labs. One is in Osaka and one in Riken. And you can see many young people are in my lab. So before moving to today's main talk, I would like to briefly introduce what's ILC2. So ILC2 is characterized by the high cytokine producing capacity, and its production is independent of antigen stimulation. So when we identified this cell, we only knew that ILC2 produce IL-5 and 6 and 13 and 4 and also GM-CSF. But later, it had been clear that ILC2 produce many other cytokines, which I'm showing here. And the role of these cytokines had become clear. So recently we reported that GM-CSF controlling the balance of bone metabolism, and filigrein have been reported to act in the tissue repairing. And both IL-5 and L-taxin is important factor for induction of eosinophilia. And also, I'm going to talk this later, but IL-5 is very important factor for B1 cell self-renewing. And IL-6 can support antibody production from B cell, and IL-13 induce goblet cell hyperplasia and induce mucin production or epithelial cell regeneration. And I also going to talk about this a little bit later, but IL-4 from ILC2 can induce low affinity IgE production from B1 cell. And both IL-2 and 9 have an autocrine effect, and both of cytokine support the survival or proliferation of ILC2. So to talk about the discovery of ILC2, I cannot ignore this guy, Shigeo Koyasu, who support me a lot on this research. So after I graduated from dental school, I joined to Shigeo's lab at Keio University in 2005 to study basic science. And Shigeo was the one who reported the importance of PI3 kinase in immune cell. And most members of his lab were working on PI3 kinase at that time. So, when I joined to his lab and my first day in his lab, he said that the delicious PI3 kinase cake has already been devoured. So find your new project by yourself. So you might you can imagine how I was shocked with this comment. But so I was like this for half year. But during those 6 months, I didn't just sit at my desk and reading the paper, but I try my best to do various experiments to see if I can find something, some phenomenon I can get interested in. So finally, I got two questions, and one was why all lymph nodes are embedded in adipose tissue. So as you can see here, I'm showing 6 different lymph nodes, but all of the lymph nodes are covered by adipose tissue. And when we need to get the cell from lymph node, we need to remove these adipose tissue first, right? So I expected that adipose tissue may have immunological role, which we don't know yet about. And the second question was, how do cell transfer into the peritoneal cavity migrate systemically? So generally, IV injection is more efficient for the transplantation, but cell can be spread systemically even by IP injection. So when I learned this, I wonder how the cell transfer by IP injection would migrate out from the peritoneal cavity, because no vasculars are opening into the peritoneal cavity. So I expected that cell in the peritoneal cavity may have unknown migration route. So these two questions led me to do this ridiculous experiment. So in this experiment, I directly inject 1% Triton blue, which is a blue dye, into the peritoneal cavity, and to see where the this blue dye gonna leak from. So, but as a result, all of the tissue in the peritoneal cavity was stained in blue. So I thought I failed this experiment. But when I look the mesentery under microscopy, I found that there are some spots which stained by this Thuringian blue. So I wonder what they are, and that's why I made a HE section of the mesentery and went to the pathologist office and asked, what is this structure? And that guy said, oh, Kazuyo, there are so many such small lymphoid clusters in our body, and it's not uncommon. So I think you can imagine how I was deeply disappointed with this comment because I was looking for my team for PhD. And so I disappointed and go back to lab and start writing a notebook about this result. But I suddenly realized that I had forgotten to ask him the correct name of this structure. So I went back to his office and asked him about the name, and he said, oh, Kazuyo, no one put the name on such small structure. So that's why we later term that this structure is fat-associated lymphoid cluster, FALC. Okay, so next I wonder what kind of cell exists in the FALC. So I made this as a PhD question. So this is a copy of my lab notebook from the day I discovered ILC2. And first of all, I must explain a little bit about the mouse, which I used for this experiment. So many, if people working on PI3 kinase, you might think I was looking something for PI3 kinase because I use P10 lithium Cure mice and also PI3 mice, but this is without Cure, and this is PI3K plus minus, which is almost like wild type mice. So since I had already been Shigeo's lab for 10 months and research progress was not going well compared to other students, I was too sorry to ask Shigeo to buy me a new young mice for my preliminary experiment. So I was always using such old mice which other people no longer needed. But I was lucky enough to use these old mice because the number of ILC2 increased by aging. So it was easy to find out this population in old mice. So here, I think you can see 90% of cells are expressing c-KIT and SCA1. So I staying with c-KIT and SCA1 and Fc epsilon receptor alpha. But I found that this population is missing in biopsy background mouse, and I wonder why. So if there's no difference between B6 and biopsy, I think I couldn't find out this population, but this time fortunately, I could notice there is population here, but not here. And so later I learned that SCA1 expression in biopsy mice is very low. So, and also c-KIT APC, which I use in this experiment, is not a really good antibody. So if I use c-KIT like PE or P7, I can bring the ILC population here. Anyway, this difference let me know we're here. So in this experiment, I wrote some comment here in Japanese. Let me translate in English. So I wrote, it is interesting that there are many c-KIT SCA1 cell in the B6 mice. So this means that I did not realize at that time that this was the moment of the discovery of ILC2. Anyway, I was happy to find some unique population in the mesentery. And later, I found that this c-KIT SCA1 double positive cells are negative for lineage marker. I'm showing lineage marker here. And we confirmed that c-KIT positive cell is exist abundant in the FALC structure, but not in the other fat, like adipocyte area. So let me tell you that success doesn't come easily. So beginning of the nightmare days, linear c-KIT SCA1 cell. Okay. So what's lineage negative c-KIT SCA1 positive cell? So if you search this phenotype in PubMed, you can find many reports about hematopoietic stem cell. So I strongly believed that c-KIT SCA1 cell in the mesentery must be the stem cell or progenitor. And from 2006, February 16 to 2008, February 18, for like 2 years and 70 days, I did 385 experiment on like a developmental analysis, because I strongly believe this should develop into some mature cell. But at the end, at like experiment number 520, I wrote the comment here that I have performed many transfer experiments in third of these experiments, lineage negative, c-KIT SCA1 positive cell differentiated into T cell. So I have believed that these cells are T cell progenitor, but this may be just a contamination. And it is also possible that this cell are already mature cell and just expressing c-KIT and SCA1 as a marker like mast cell, because everybody know that mast cell express c-KIT as a marker. So now I'm ready to assess something about the cytokine reactivity of this lineage negative c-KIT SCA1 cell. So from this experiment, what I can say is, if you get stuck in your experiment, try to getting away from the old idea and consider it from completely different angle. So once the idea allows that lineage negative c-KIT SCA1 cell might be a novel cell, my experiment began to work like a flowing liver. Because by the Giemsa staining and electron microscopy image, we can say that this population is belonging to lymphoid linear cell with circle shape and big nuclear. And we also confirmed that this population exists in wild type and RAG2 and nude mice and allele mouse, but lacking in common gamma chain knockout mice due to the lack of IL-7 signaling, which most of lymphocytes require for the survival and the development. And also we confirmed that this population exists in the ROR gamma deficient mice, which means that this is not same as ILC3 population. But looking in ID2 knockout mice, which most of innate lymphocyte is missing. And we also confirmed the phenotype of this population, and the phenotype looks very similar to T cell because it's CD45 positive and expressing IL-7 receptor and positive for Cy1. And the activation markers which T cell express was also expressing this lineage negative c-KIT scorn cells like CD27, 38, 25, 44 GITR, and CD69. And the most significant finding of this study was that microRNA analysis revealed that these cells express high level type 2 cytokine like IL-4, 5, 6, and 13. And we also found that co-stimulation of IL-2 plus IL-25 induce a huge amount of type 2 cytokine like IL-5 and 13. And if we compare this amount of cytokine with IL-33 stimulated ILC2, the amount will be like this because IL-33 strongly induce this cytokine production from c-KIT scorn double positive cell. So 2008, December 13th, at the year end, we wrap up these new finding on and we submit our paper to Science, okay, which was my favorite journal. And 1 week later, it get rejected. But I was young enough to say, Shigeo, that we should never give up. Okay, so the first paper was first paper we submitted to Science did not reveal anything about the function of lineage negative c-KIT common cell. So we decided to investigate the role of this cell in steady state and in inflammation. So for the steady state role, we focus on the low IL-5 constitutive production, because compared to IL-13, ILC2 can produce IL-5 constitutively even in the naive phase. So at that time, we knew that for the self-renewal of B1 cell in the peritoneal cavity, IL-5 is a very important factor. And but people believed that the source of IL-5 is T cell. But T cell cannot produce IL-5 without antigen stimulation, which means that it might not be T cell who are producing IL-5 for the B1 cell. So we isolate B1 cell from peritoneal cavity and labeled by CFSC and co-injected with PBS or CD4 T cell or c-KIT scorn double positive cell, and wait for 9 days. We use common gamma RAG2 double knockout mice, which locking all lymphocyte, and assess by FACS for the cell division. So the number of B1 cell is shown like no division once or twice or third in different color. So this data suggests that T cell cannot accelerate the self-renewing of B1 cell, but by IL-5 production, c-KIT scorn double positive cell can support the self-renewing of B1 cell. And for the role of c-KIT scorn cell during the inflammation, we decided to use the Nippon-Strangers sporogenesis infection model. So it was known that during this Helminth infection, a huge amount of IL-33 are produced, and also IL-4 and 5 and 13 production occur. But no one knows that what kind of cells are producing such cytokine during this infection. So we confirmed that IL-5 production and IL-13 production is occurring in both wild type and Rab2 deficient mice, looking T cell, B cell. But these productions stop in common gamma chain Rab2 double knockout mice, which mean that ILCs are important cell for the production of IL-5 and 13. And it was known that goblet cell hyperplasia occur by IL-13 production. So you can see white spot on the epithelial cells here and here, but not here. So because IL-13 production is missing here. But when we, so we confirm the IL-33 level in the peritoneal cavity. And when we inject c-Kit skull double positive cell into the peritoneal cavity during this infection, we see the recovered goblet cell hyperplasia, which means that c-Kit skull double positive cell can respond to IL-33 during the Helminth's infection and produce IL-13 and induce a goblet cell hyperplasia. So again, we wrap up our result, and on like May 28, 2009, we send it to Science, but rejected again. So now I was also young enough to give up easily, but our paper was published in Nature because Shigeo didn't give up and send our paper to Nature 1 week after we rejected from Science. So after 3 revision, our paper was accepted in November 5th, which is my birthday. So that's why I want to show this process. So I was really happy, and in that paper we termed this c-Kit skull double positive cell as natural helper cell, and but 3 months later, Andrew's group report the cell name termed myocyte, and also David's group also published the paper about MPP type 2. And so we were wondering why Nature accepted our paper. Even this paper was rejected twice by Science, but now we know that the Nature Press was handling 3 similar paper at that time, and know that some new Nobel type 2 cytokine producer will be reported soon. So we were so lucky. But anyway, also I think both natural helper cell and myocyte is quite similar cell, but MPP type 2 cells was reported to have a potential to develop into the myeloid lineage cell. So this might be a little bit different from our cells. Anyway, we again termed this cell as group 2 innate lymphoid cell. And now this naming is more common to call this cell type. So definition of ILC2 is lineage negative and GATA3 positive. And also we need to check the type 2 cytokine production ability under antigen independent manner, because this is very important function of ILC2. And some people try to identify ILC2 by like cytokine receptor, like IL-33 receptor or IL-25 receptor, but this is not good way to detect the ILC2, because sometimes, I'm going to talk this later, but sometimes ILC2s do not express IL-33 receptor or IL-25 receptor. So 825 is the number of experiments that I performed for this paper. So what I want to tell young people is Your desk will never give you the answer, but your bench will tell you the answer. Okay? So with the discovery of ILC2, the concept of innate lymphoid cell subset have been established. So ILC2 population closely mirror the helper T cell subset in their cytokine profile. But while T cell require antigen stimulation for cytokine production, ILC2 only requires cytokine stimulation for the cytokine production. And both the helper T cell and ILC2 have similar role in the disease. So both Th2 cells and ILC2 produce IL-4, 5, 13 and act in the parasite infection or in induction of the allergic diseases. And now people know that ILC2 distribute in variety of tissues showing here. So the number is quite a lot in adipose tissue or lung or intestine and skin. But I can say that ILC2 exists everywhere. So Junichi, who was my first, not my, Shigeo's student, was I was doing research with him, focus on the signaling pathway of ILC2. So he wondered which of the pathway under IL-33 receptor is important for the cytokine production or development of ILC2. So he used 3 different inhibitor for NF-κB pathway or 2 different MAP kinase pathway, and clarify that SP, which is an inhibitor for P38, can clearly stop the phosphorylation of GATA3 and also stop IL-5, 6, 13 production of ILC2. So P38 GATA3 pathway is very important pathway for the cytokine production and differentiation of ILC2. We also assess how ILC2 can go in and out from the tissue, but the result was what we didn't expect it, because we found that ILC2 are tissue resident cell. So in this experiment, we made parabiosis mice with B6 mouse and B6 estrogen mouse. And in parabiosis mice, the blood circulation is shared. So here you can see after like 1 month, T cell gonna be mixed in 2 different mice, but ILC2 didn't mix, which means that ILC2 cannot easily move to other tissue. And this is one of my favorite report published by Richie's lab. And they isolate ILC2 from bone marrow, fat tissue, lung, and gut, and skin, and compare these 5 different tissue-derived ILC2 by single-cell RNA sequence analysis. And this data really showing very important thing, which we should remember, because all of the ILC2 population expressing GATA3 and IL-7 receptor, but IL-33 receptor expression is limited in fat and lung and bone marrow ILC2, and missing in skin or gut ILC2. But IL-25 receptor is highly expressed in the gut-derived ILC2. And they also reported that the source of IL-25 is in the intestine called tuft cell. So I think intestine can provide a good environment for ILC2 by this IL-25 expression and receptor. And skin have very unique population because they do not express IL-33 receptor or 25 receptor, but express IL-18 receptor. Okay, so Satoshi, who was PhD student, says how does ILC2 develop? So people in this field first thought that bone marrow progenitor is the progenitor for mature ILC2 in the all tissue, but we thought that ILC2 is tissue resident and it should come to these peripheral tissue during the fetal stage. And Satoshi found that in the fetal stage, we can detect ILC2 like in this gate. So here to here is the fetal stage, and this is neonatal. But we can detect this IL-33 receptor, IL-7 receptor positive, lineage negative, ILC2 even in the fetal stage. And what he focused on was this population, which is negative for T1ST2, but expressing IL-7 receptor. And he hypothesized that this population might be ILC progenitor. So he isolates stromal cell from mesentery and divided in 3 different subsets. GP38 positive and PDGF receptor positive and double negative, and co-culture the stromal cell with this progenitor and see the development of ILC2. And he found that PDGF receptor alpha positive stromal cell can support the differentiation of ILC2 from progenitor to mature ILC2. So this means that all of the, and he also confirmed this in the lung, and so this means that ILC2 first developed from fetal liver, and during the fetal stage, they migrate into each peripheral tissue, and in those tissue, the specific stromal cells support their differentiation, maturation. So I think the discovery of ILC2 has had a major impact on the field of allergy, because before ILC2 was discovered, people in the allergic research believe that Th2 cells are the cell which develop the allergic reaction. So as I'm showing here, when allergen come into our body, dendritic cell take the portion of this antigen and bring this into the lymph node. And in lymph node, this antigen stimulation gonna induce antigen specific Th2 cell, and this gonna migrate back into the inflammatory site, and in response to this specific antigen, Th2 cell produce type 2 cytokine. But what the people in this field clarify was this is one type of allergy, but there are different pathways to induce the allergic reaction by ILC2. So in this case, allergen are required, but none of cell recognize this antigen directly. But we now know well that many of allergen have a protease activity and kill the epithelial cells directly. And these necrotic cells gonna produce IL-33. And in response to IL-33, ILC2 produce type 2 cytokine during the allergic reaction. So now we think that there are 2 different kinds of allergy. One is antigen-dependent allergy, and the other is IL-33 dependent allergy. So for the development of allergy, genetic background is very important, and people are well known about this. But for like a peanut allergy or cat allergy or like pollen allergy, which patient can realize what kind of allergen they are reacting, should be called like a antigen dependent allergy. But like fungus or ticks or like a fruit related allergy is, I think, mixed type, because these things can be recognized by like recognized as an antigen by T cell, but also they have protease activity and can induce IL-33 production, and as a result, ILC2 will be activated. And these things like air pollution or cold exposure or exercise is known to induce allergic reaction, but no one knew what kind of cells are inducing this allergy. But recently, 3 of them were reported to produce IL-33. So I think ILC2 are related to this kind of allergy. So stress-induced and medicine-induced is still unknown field. So to assess ILC2 in human sample, we collaborate with Yoshitaka, and because he had very good technology to detect the cytokine production from at the single level. So as you can see here, we see the cell in like small well, and under microscopy we can see IL-5 production in color red and IL-13 production in color green. So we isolate ILC2 from both healthy volunteer and allergic patient and compare the cytokine activity of ILC2. And here, the data was very beautiful because none of ILC2 from healthy volunteer show IL-5 or 13 production, but like more than half of ILC2 from allergic patient show very high activity of ILC2. So many people try to assess the importance of ILC2 in human by counting the number of ILC2, but I think that's not good idea because many of patients have already getting the treatment, and sometimes those treatment like storage treatment going to decrease the cell. But if we carefully look on the cytokine activity, we can say this cell is still activating and this cell is not. So I think this technique is a very useful technique in the future ILC2 research in human. So Hiroki was PhD student and also he's clinician. So his clinical question was, how does a severe asthma develop? Because the mild asthma, the difference between mild asthma and severe asthma is the steroid resistance. Because for most of normal middle class asthma, steroid treatment is effective, but in the severe asthma patient, it's not. So he wondered what is happening in our body. So he found that IL-33 stimulated ILC2 can be suppressed by normal steroid treatment like dexamethasone treatment. But when ILC2 got the dual stimulation by TSLP plus IL-33, as this data suggests, even this is under dexamethasone, the number of ILC2 get increased and IL-5 and 13 production will occur, which means that TSLP are gonna activate STAT5 and induce the BCL-XL expression, which is an anti-apoptotic molecule. And that's why ILC2 become steroid resistant and induce a severe asthma. So I think that this was really beautiful work. And now this what phenomenon is confirmed in human ILC2. So I need to thank Miho and Natsuki, who are my technician, for big help for this work. So in this paper, we focus on the suppression mechanism of ILC2 because it looks ILC2 is really doing bad for allergic reaction. So what we found was interferons can suppress ILC2 and interferon gamma is the strongest suppressor for ILC2. So if we inject IL-2 plus 25 or IL-33 in the tracheal, we can induce ILC2 in the valve, and also we can see a huge amount of eosinophil in the valve. But if we co-inject interferon gamma with the cytokine, ILC2 will not appear in the valve, and the eosinophilia are going to be stopped. And also we measure the cytokine in the valve, and IL-5 and IL-13 production was clearly suppressed by interferon gamma. And I published this paper with Hiroki, and he was an equal contributor in this paper because he also found that IL-27 can suppress ILC2. And so we compared IL-27 receptor expression in T cell and ILC2, and he found that this receptor expression is extremely high in ILC2. So therefore, during the Alternaria-induced asthma model, IL-27 couldn't stop the activation of Th2 cell, but clearly stopped the activation of ILC2. So now we know that both interferons and also IL-27 are the strong suppressor for ILC2. So Takaharu did very unique experiment on like obesity. So he found that when we fed the mice with high-fat diet, somehow common gamma RAG2 double knockout mice which lacking ILC subsets will not gain their weight compared to wild type or RAG2 knockout mice. These mice are fed by normal diet, and these three were fed with high-fat diet. But as you can see here, common gamma RAG2 deficient mice show resistant to the obesity. This is a picture of liver. So RAG2 knockout mice, common gamma RAG2 double knockout mice show very healthy liver even after high-fat diet feeding. So he hypothesized that ILC2 might be inducing this phenomenon. So he first inject ILC2 from adipose tissue, and but unfortunately we couldn't see any recoverment of weight gain, and he put this experiment away for like 2 years. But one day he was at that time working on intestinal ILC2 and told me that there are some extra intestine derived ILC2, so he wanted to do this experiment again with this intestinal derived, gut derived ILC2. So I said yes, and he was really surprised with this data because we couldn't observe any increased body weight by fat-derived ILC2, but somehow small intestinal derived ILC2 could recover the body weight gain, which means that there are so many ILC2 in the body, but each tissue-derived ILC2 have a specific role. And for obesity, small intestine derived ILC2 is doing something important. So recently, Yuki have reported a very beautiful paper about transcriptional regulation of ILC2. And in this project, she focused on TTP, which is quite major transcription factor, which degrade the messenger RNA. So what she found is during the steady state, IL-5 messenger RNA, which related to the constitutive IL-5 production, will be degraded by TTP. So that's why ILC2 can just produce small amount of cytokine. But in TTP knockout mice, this degradation gonna be stopped, and ILC2 will have more messenger RNA. So we could observe enhanced IL-5 production from ILC2 and induce more eosinophilia. And in the IL-33 dependent inflammation, we well known that IL-33 receptor directly induce the phosphorylation of P38 and induce GATA3, and that's why ILC2 can produce a large amount of cytokine. But during this reaction, TTP gonna be disappear by IL-33 stimulation and contribute to this excessive IL-5 production. So I'd like to a little bit show some ongoing project in our lab. So Yasu is focusing on IL-4 production from ILC2. And he clarified that IL-33, single IL-33 stimulation is not enough to induce IL-4 production from ILC2. But if we stimulate ILC2 with IL-2 plus IL-33, we can induce IL-4 production. And what he's focusing on is this IL-4 production from ILC2 can induce low affinity IgE without antigen stimulation. So in this experiment, he also inject IL-2 plus IL-33 intranasally and detect IgE in the serum. So this data was quite surprising data for me because I thought IgE production only occur by antigen stimulation, but without antigen stimulation, we can induce the class switching of IgE. So he wondered which cells, which of B cell, B1 or B2 cells is producing this unique IgE, and found that B1 cell, shown here, is the source of this low affinity IgE. So we're now calling this unique IgE as innate IgE because this IgE do not require antigen stimulation for the production. And he also assessed the role of innate IgE in this experiment. So if we inject papain into the trachea, we can induce mast cell and basophil and ILC2 and eosinophil accumulation. But when we do this experiment in IL-4 knockout mice, which also lacking IgE, the number of these cells gonna decrease, right? But if we pre-treat these IL-4 deficient mice with innate IgE, the number of mast cell, basophil, and ILC2 and eosinophil will recover, which means that innate IgE is involving the susceptibility of the allergic reaction. So we're still working on this and would like to publish soon. And Natsuko, who is also a PhD student, focus on the pulmonary fibrosis. So the reason why we start focusing on pulmonary fibrosis is because when we clarified, we report that interferon gamma suppress ILC2, and my friend Kazuyo Moro's group reported Treg can suppress ILC2. We established the mouse called interferon gamma receptor RAG2 double knockout mice, which lacking both suppression mechanism on ILC2. So we expected that this mice will develop more severe allergic diseases, but what we found was this double knockout mice spontaneously developed the pulmonary fibrosis. So it was quite surprising data. So the number of ILC2 increased by aging in this double knockout mice. So onset of this pulmonary fibrosis is around like 16 weeks old. So at that time, ILC2 increased. So we made a triple knockout mice with common gamma chain knockout mice and confirmed that without ILCs, pulmonary fibrosis will not be developed. So next question was, which of ILCs are important for the onset of this pulmonary fibrosis? So we made IL-33 and interferon gamma RAG2 triple knockout mice again. And this data suggests that ILC2 in ILC subset are the most important cell for the onset of fibrosis. So she confirmed that ILC2 can directly induce the collagen production from fibroblasts by co-culturing the ILC2 with fibroblasts. So without IL-33, ILC2 could not induce collagen production from fibroblasts. But with IL-33 stimulation, it turned in red color, which means that collagen production is more in this well. So this means that ILC2 can directly induce collagen production from fibroblasts when they get activated by IL-33. So everyone in this fibrosis field know that fibrosis occurs first. First step of the fibrosis is inflammation, and then change it to fibrosis. But it might be a little bit difficult to see, but we stain ILC2 in red color and confirm that ILC2 appear in the inflammatory region first before the fibrosis occur, and it's still abundant in the fibrotic region. So if I make this picture bigger, you can see that ILC2 and fibroblast is directly connecting. So finally, we confirm if ILC2 is important cell in the human idiopathic pulmonary fibrosis patient or not. So we collect the ILC2 from healthy 10 healthy volunteer and 20 IPF patient, and this principal component analysis show that a patient ILC2 in the patient is very different from the ILC2 in healthy control. And we also confirmed that ILC2 related cytokine and receptor expression is increasing in the ILC2 from IPF patient. So this is IL-33 receptor, and SPP1, which is a very critical factor for the fibrosis, was also increased in this IPF patient. And as same as our spontaneous fibrosis knockout mice, interferon gamma receptor was strongly decreased in the IPF patient, which means that similar mechanism is occurring in the IPF patient. So in summary, I'd like to end my today's talk with this picture. So ILC2 are regulated not only by cytokine, but also lipids or neuropeptide or hormone. And although immunology has developed along with the study of antigen specificity, I believe that more attention should be paid to antigen independent cells such as ILC2, since disease is regulated not only by antigen but also by a variety of endogenous factors. So there are so many people who contribute to this project, and I cannot put all of them's name here. So I would like to show my sincere appreciation with this picture. So I would like to thank Shigeo, who really support me for a long time, and believe my project will be interesting for like a first 5 years. And so we moved to RIKEN at 2012. And the member become expanded on 2013 because I got the position in Yokohama City University, and many students joined to my lab, and I was also belonging to Shigeo's lab. So both members worked together at that time, and I become independent from Shigeo's lab on 2015, and now half of my lab member are working at Osaka University, and half are working at RIKEN. So I would like to thank all of them for huge support, and I always love to discuss with them. So Laura, that's it, and thank you.
Thank you.
Should I stop this? Okay.
That was so wonderful, Kazuyo. It was so wonderful to see your laboratory's incredible body of work. And what I think is going to be so inspiring for all the young scientists to see is your incredible journey.
So too.
And all the lessons you learned, and seeing your notebooks through to the 825 experiments. It's really showing that these things, they do not come easy. And it was just such a thrill for you to take us on that journey. It was truly amazing. I am just going to share my screen just to let everybody know how to ask questions to Kazuyo, who will be happy to answer your questions on Twitter via the Q&A, and that will be available for some time after the talk. So please put on your questions when you have them, and kindly just hashtag in Global Immuno and reply to the tweet that says "Ask questions for Dr. Kazuyo Moro here." So that leaves me to remind you, same place, same time next week, we will be hosting Ansu Sampathy from Stanford University. So please tune in for that. Again, on behalf of all of us around the world that are watching these IMMUNOTALKs, thank you again so much for your wonderful, wonderful, inspirational talk. We have so appreciate it. And that leaves me to close the session of Global IMMUNOTALKs. Thank you.